Hi everyone, I'm Charlotte Kilpatrick reporting for Vaccine Nation from the World Vaccine Congress in Washington. I'm delighted that today we're joined by Renee Hart of LumaCyte. Thank you so much for making time for us.
Happy to be here. Thank you.
No, thank you. So to kick us off, would you kindly just introduce yourself and the role that you play within the organization?
Absolutely, absolutely. So I'm the president and co-founder of LumaCyte. LumaCyte is a novel, label-free, single-cell analytic used in vaccine development and production.
Fantastic. So you joined us for a roundtable yesterday, I believe?
It was,
Fantastic.
yes.
So this explored the importance of viral titers. Could you elaborate a bit on why a real-time viral titer is so important to vaccine manufacturing?
Absolutely, absolutely. So historically, and even currently, when a vaccine is manufactured, they have to rely on historical averages for the TCID50 or the viral infectivity analytic. And the challenge with that is that every vaccine production run is slightly different. And so the viral titer could be higher, it could be lower. And so it does result, with the current analytics, it does result in some out of specification or batch failure events. And with a real-time viral titer, being able to pull a sample directly out of a bioreactor, run it on our Radiance laser force cytology machine, platform instrument, you are getting a real-time viral titer in 5 minutes. The power of that real-time viral titer is really transformational because you now understand where in your manufacturing process your viral titer is. So before you begin scale up or during scale up, you can assess the situation. Do we need to seed more? Do we need to dilute before we scale up to the additional larger vat? And that improves the consistency of the manufacturing, and that is really a big focus for the FDA right now. There's a lot of draft guidances being released. Last July, there was the CMC draft guidance by the FDA for CMC manufacturing for advanced therapies and vaccines. And so this technology does support that because you now have that opportunity to implement a live PAT technology, process analytical technology, that connects the biological evidence to the analytic through a real-time viral titer, specifically for vaccines. And the other real power in that is not only are you getting a more consistent production run, but you also are able to dose the vials more appropriately. So not relying on historical averages, you will understand what the exact viral titer is, which allows you actually to make more doses with the same cost of goods, the same batch material. So that's really powerful. The third reason that it's really important is that when you know that viral titer and you understand the accuracy of that viral titer in your production, you actually can predict and extend in many ways your shelf life. So you're not relying on assuming the lowest viral titer and how it degrades over time for that shelf life. You actually have the exact viral titer, so you can actually get more shelf life out of that product. Really powerful for pandemic preparedness.
Fantastic. So you've sort of given us examples of how these solutions are really changing the game for the vaccine community.
Yes.
Could you tell us a bit more about the innovation that you're bringing to this community?
Yes, absolutely. So laser force cytology is a novel new technology. We've launched our commercial instrument back in 2019. The technology laser force cytology has been developed over the last 24 years, and lots of publications, lots of collaborations that we have with regulators, and we're really being seen as a forward-looking technology that can really transform a lot of the challenges in today's landscape for not only vaccines but also advanced therapies. And one area that we didn't really realize was so powerful that we do is comprehensive cell characterization. So what that means is when you start a manufacturing run, whether it's vaccines or in CAR T and cell therapy where you're trying to understand the donor material, the cellular donor material, being able to comprehensively understand what that cell is today, but what it will look like tomorrow. Meaning, is it going to be strong enough to proceed with the manufacturing for that product, for the specific patient for a cell therapy, or for the vaccine production, right? If you don't have a healthy, strong, robust cell that can be strong enough to manufacture the virus, to produce it in the vaccine manufacturing process, you're going to have lower yields, right? So that's another, I guess, a fourth reason in that first question. You do get higher yields, right, with understanding that viral titer throughout from early process development all the way through QC. So just for us to be able to not only bring that additional cellular comprehensive characterization knowledge to the developer and the manufacturer, that has profound impact. And so we've been told that is a very big innovative solution. And one thing I'll also share that's new for us and just in general for this application is we recently showed 3 weeks ago that we could correlate the cellular starting material of the PBMCs for patients in CAR T and correlate that to the killing assay, the co-killing effect of those patients. So your cells may have a really high killing efficiency where my cells might have a lower killing efficiency. So being able to know that up front is really powerful for those type of treatments.
Amazing, that sounds really exciting. So that leads quite nicely into the next question about the space you're filling at the moment. Are you meeting an unmet need that you've identified, or are you sort of setting yourself apart from others in this space?
Yes, thank you. I would say we're filling an unmet need in a couple different areas. We were very lucky and blessed in many ways to be invited by the FDA to train all of their vaccine and cell and gene therapy reviewers. So in January of 23, we trained 120 FDA reviewers. And in that discussion and in that training experience where they've gone through a lot of our data and kind of suggested different case studies for their training, they realized the power of the technology for cell health, and that's where that came from. They asked us to really focus on that type of solution for the industry because they said that the industry doesn't realize the challenges around, I guess, a faulty understanding of cell health, meaning if you just get a viability check mark and off you go, that's only an instantaneous view of how that cell looks at this minute. Where 4 hours, 8 hours later, it could already be heading towards apoptotic trending, but the scientists wouldn't know it. And so we hadn't realized the power of our technology in that application. And so we were told that if we can predict the success of the cellular material and any of those applications I just went over earlier, that it could have profound impact in the success of the manufacturing and success of the efficacy for the treatment. So that's been really exciting for us, to really walk down that path. And I'm also excited to share that as part of that, we were invited into a long-term CRADA with NIST. And NIST is working very closely with us and running a lot of samples in their labs on radiance for cell fitness and better characterization. And they've presented at quite a few conferences recently, and they'll be presenting at International Society for Cell Therapy later in May. But they did a lot of really unique things where they're pressure testing the technology, laser force cytology, where they're adjusting the glucose concentrations, they're adjusting the pH, adjusting temperature, very subtly, 20% up and down, and just to kind of see what we see versus viability. And viability is showing positive, like green check mark, but we're showing those apoptotic trending. So really important, because if you are, the world is, freezers go down, the temperatures change, your media conditions change, or maybe somebody mixed things differently, right? Those little changes can have a really big impact. So if you could understand that up front with your analytic, like immediately at the beginning, you wouldn't waste all that time going down the assay. A lot of wasted resources, a lot of wasted material, and time, getting life-saving treatments and cures to patients, in need. So that is one, area I think that's was eye-opening to us, where we really are serving an unmet need. The other, I guess, is the real-time viral titer. We are a replacement for TCID50. So for the vaccine community, that's a really powerful thing because it's a painful assay. I think it's 70 years old, maybe older. It's a very old, antiquated assay. And the FDA is very excited about something that is easy, it's precise, it's accurate, and we're automated. So you're taking the labor and the hands-on work from scientists away. You're putting a well plate in, clicking play, and getting your viral titer in 5 minutes, for a production run. So it's really transformative, from that perspective as well.
Absolutely. So it sounds like you have brought a lot to the Congress in terms of sharing all of this information and the solutions you're bringing to the community. Can you tell us a bit about how your experience of the Congress has been so far? What have you learned or achieved in your time here?
Yes, it's been a really nice conference. It always is. I'm proud to say that LumaCyte has been attending the World Vaccine Congress since 2015, and it's amazing to see the growth. It really is. I mean, I think we're just shy of 4,000 registrants this year, and I think in 2015 we might have been around 200. So we've grown so much, which is just really exciting to see. For us, last night was really special. We were part of the VIE Awards, and we were nominated and then became a finalist, notified. We were very surprised. We didn't know at all, but we were a finalist for the best process development and production technology. And then last night we found out that we were highly commended, which is second place, runner-up. Out of the 8 finalists, we got second place. So we were just really proud and honored to be recognized by the community and the industry. And I would say, for me, like, one of the real nice takeaways was listening to Peter Marks yesterday in the plenary session, where he talked about we need novel cellular analytics. We need to implement these because we're not going to get by with the status quo. And so it's just really exciting when you see regulators, and very important regulators such as Peter Marks, stressing the importance of cellular analytics. And that's what we're just proud to do our part. The community is a very special community. It's a hardworking community. They care deeply about the patients in the world, and we're just thrilled to be part of it. We're just one small part. They're doing the amazing work, and we're just giving them a tool, that can help them do that work better. So it's just great to be here. It's great to be part of the excitement in the industry. I love the focus on oncolytic vaccines. I mean, can you imagine if we get to a stage in our world where we don't have to deal with cancer, that we can prophylactically treat that? It's transformative.
Thank you so much for your insights. It was a real pleasure to speak to you.
Yes, you as well. Thank you, Charlotte.